right, good afternoon. My name is Rashid Bashir. I'll give you a quick overview of activities that I've been involved in. So just very briefly, I thought some of you are new here. This is actually just a slide about this building. So this is the Micro Nanotechnology Lab. This is one of the interdisciplinary research units on campus with the goal of advancing the state of nanotechnology, essentially. We have a clean room in the building. That's the largest clean room on campus. So if anyone needs to use clean room for the kind of work they want to do. Also, we have a Bionanotechnology lab, which is available also on a charge per use basis. The building actually has about 220 residents, which includes students, postdocs, and lots of users of the facility. There's been a lot of activity in the last few years with the hard work of a lot of faculty, Brian, and many others. And there's a lot of work on electronics and photonics in the building, which are many ECE types. And then a lot of work on integration of nanotechnology and medicine, which is clearly this group is one example of that. So I just wanted to throw that out very quickly. And then just two slides I want to show real quick before I talk about some of the research things. Just to also let you know, we talked about student training earlier. So we're really firm believers, of course, that that's, of course, what we do for a living in terms of training students. But we also have two training centers and training grants. One is an NCI training grant on cancer nanotechnology. So a lot of actually our colleague JJ is part of that, Brian, myself. And this is funded by NCI. So we have about 25 faculty from around campus that we have pulled together looking at nanotechnology for cancer. So again, in terms of an ecosystem and in terms of having these students then exposed to industry, we really see CABPN as serving a great purpose for us. And you'll see many of the students tonight, actually. We also have an SF IGERT on cell mechanics and nanotechnology. So this is looking at, we know that cells communicate electrically, we know cells communicate chemically, but in the last 10 years or so now it's also been established that cells communicate with each other mechanically. So the mechanotransduction and the physical forces are very important. So there's a lot of mechanical engineers and biologists that are part of this effort. And this funds student trainees and NSF, of course. Yeah. In my group, just to also give you a quick, that's what Brian had asked, we're interested in using various fabrication methods in silicon and MEMS and polymer PDMS to really apply that to grand challenges. So the two projects that I'll talk about tomorrow are actually both in this space right here in the diagnostics for food safety and global health. I won't mention those right now, but just right now I'll talk to you about a couple of other projects we have. So we have an active project funded by NIH and NSF on developing point-of-care sensors for detection of microRNA and other proteins that might be of interest for cancer. So we actually have a technology where we use basically silicon chips. We make these top-down fabricated nanowires using a VLSI-compatible process, and then we attach biomolecules on the surface of these nanowires or nanoribbon structures using various surface chemistries. And then we can detect microRNA, for example, which are short nucleotides. And now it's been established that microRNAs are very important for cancer. There's no commercial or there's no actual test out there yet, but it's now pretty established that microRNAs could be important markers for cancer. So this is a label-free electrical method for detection of these biomolecules, and we've been using it for proteins, but now really focusing on microRNA. So we apply the same silicon nanoribbon or nanowire technology also for heating of droplets, which was a project funded by CABPN. And actually this project also has an industry partner, which is TSMC, and now we are working with them to actually get the silicon chips fabricated so that we don't have to do that in our fab itself, because that just takes a lot of time also. We're also very interested in using other types of nanosensors. So this is another project to look at cancer per se specifically, but this also has applications in DNA sequencing. So this whole idea of using a nanopore, so we are working on making solid-state nanopore channels for detection of DNA and DNA protein complexes. We have processes that we have developed using nanofab films that we can deposit, these atomic layer deposited film of aluminum oxide and also now hafnium oxide. We can use then an electron beam to drill pores through the single pores. So this right here is a 5 nanometer pore through a membrane. So the idea is to mimic a lipid bilayer with a protein channel, but make it with a solid state device. And then we can drive DNA through this using voltage, and the idea here was to use a methylation binding domain protein, these proteins that bind to methylation sites on DNA, and pass that complex through this nanopore to potentially be able to see electrical signatures from those single molecule. So here's an example of some very recent results. This is just submitted for publication, where we have schematically shown here a DNA which will have these MBD protein bound to it on the methylation sites. You pass the complex and you'll see signatures like this of the current, where you see these various pulses with these sub-events, whereas when you pass the DNA alone without the proteins, you only see the translocations of the DNA molecules going through. But when the DNA has these proteins attached to them, you see there's more information in these pulses, and from that we can try to determine how many proteins are bound or whether proteins are bound at all or not. So the idea here is to have a technique for single molecule methylation detection. And this, of course, has applications in sequencing. We're also, along the way, we have developed a process which uses graphene as the membrane. So we have a process for using a stacked layer of graphene and dielectrics, which form the membrane, through which we drill a pore using, again, an electron beam in the materials research lab, which is another great facility we have on campus. Just briefly show that we are also interested in using mechanics and mechanical elements to characterize cells and biological entities. So one project we've been working on for some time is to try to, this is more for basic biology. We want to look at how cells physical properties can be measured, such as their stiffness or their mass. So can you measure a single cell and its mass as it's growing? So we make these resonance sensor pedestals. This is about a 60 microns on a side structure with 4 springs that's attached to a chip. But underneath it, we've actually removed the material, so this pedestal is free to vibrate in the Z direction, and then we can grow cells on them. This is all in a fluid, and we can model this using a harmonic oscillator system, but then we can also model the cell as a spring mass dashpot system, because the cell itself has a finite stiffness. It's like a jello, it's like a gel. So you can actually also extract the physical properties of the cells using this method. And then we can essentially watch single cells grow on these pedestals and measure their mass in real time and watch the single cell division. So here, 1 cell went to 2 cells, these 2 cells go to 4 cells, and so on. So you can extract some basic physical properties about cell and their growth behavior using these technologies. Sorry. And then finally, this is a project that Brian and I have ongoing too that he'll talk about tomorrow. We use, it's a busy slide, but the bottom line is that this is actually funded through an NSF STC that we have here on campus, where the idea is to build systems with cells. So as engineers, we've always built with hard materials, but this whole area of synthetic biology, and especially synthetic biology moving to cellular systems. So can we actually build systems that, not to necessarily replace an organ or a tissue, but rather think about building new systems with cells as a building block. So the current project is to take myocytes, which are muscle cells, neurons, and endothelial cells, and put them together in some interesting fashion using this 3D SLA method, for example, to develop little biobots that move, that can sense and secrete chemicals, or develop other systems that are composed of cells, essentially, cells and polymers. So there's a lot of very interesting possibilities with this. And that's it. Finally, I'd like to thank the group, my group that really does all the work. I think I finished in time.
You did? Yes, we
All right.
through a few questions.
I went through very fast. I have 2 of the projects that we'll talk about tomorrow, so I didn't mention those at all right now. Yeah.
So the 3D biofabrication piece that we talked about briefly,
Yes.
in tissue engineering is the obvious applications for that. So
Right.
can you just go into a little bit more detail about what the other
Applications.
can be?
That's a very good question. So actually, it's a project funded by an NSF Science and Technology Center, which is sort of more basic science. So the idea is to start understanding or start developing the technologies and start understanding cell-cell interaction, or how cell clusters emerge, how behavior emerges. So the goal is to then turn that into some application. So the current applications that we've been thinking about is the idea of a movable sensor. Essentially, if you have a millimeter scale structure that has cell-based systems that can move, and it senses a toxin, let's say it's moving around and it senses a toxin and it releases some neutralizing agent, something that maybe can move towards a gradient. So the application could be in vivo for something like this, or in vitro in terms of, let's say, a water supply system or something like that, where you would look for a toxin and annihilate it. There is lots of applications where cells produce specific proteins that are of interest. So can we harness that in some way with a microfluidic device that's all biological? So for example, in the center, we've been thinking about building, can you build a blood vessel? So typically, tissue engineers just replace a blood vessel, right? Can you now think about building a blood vessel that can also pump, that can sense and pump? So when the pressure builds up, it starts pumping, actually, rather than just transporting blood. So can we think about adding new functions that are not available today in biology and nature per se? But if you knew how to put them together, then you could think about things like that. So active blood vessels, cells that are sensitive to specific wavelengths of light that they are usually not. Yeah.
Thanks for your question. So I think next we have a very quick from Rashid Fargab's group. I think it's next.
Oh, sorry.